Hello, good afternoon, everyone. My name is Pedro Raposo, and I'm a curator at UniProt. And in this webinar, myself and Nadia, another curator at UniProt, we are going to talk about automatic annotation systems in our database. We're going to discuss how UniProt uses these systems to add annotation to a large number of uncharacterized proteins. So I will start with a short introduction on UniProt, and then we will look into the systems in depth. And afterwards, we will finish on how UniProt made it available for you to annotate your own set of proteins using these tools. So to start with, UniProt is the world's leading high-quality and freely accessible resource of protein sequence and functional information. We provide reliable data as we have teams of expert curators reviewing literature in order to describe what a protein does. UniProt is a massive resource as it currently has 250 million proteins that cover more than 1 million taxa in the tree of life. And we are a consortium that is composed of 3 teams, the first one being from EMBL-EBI from the UK, the Protein Information Resource from Washington, DC, and the Swiss Institute of Bioinformatics in Switzerland. And as a result, our efforts coalesce together to provide you all the services that we need in our website. So this is our website. And here you are welcomed to this homepage, where you can see in the middle a search bar to find specific proteins if you need to, or to group or a group of proteins by inserting a more general term. At the very top, you have different tools that will help you on your protein analysis, and a bit down you find the main sources that we can provide to you in our website. So these are the resources, and in this diagram we can see the interaction between them. So as a short summary, we will start by importing information externally from databases such as ENA or GenBank. And after that, we will archive those proteins as protein sequences in UniPark. And then we transform those protein sequences into UniProt knowledge base accessions, or UniProt KB accessions, where these protein sequences are going to get annotations. We also can group those proteins into different organisms, and we call those proteomes. And finally, we also can cluster those proteins together by their similarity in UniRef. But today we are going to focus on UniProt knowledge base, which is our most used resource. So the UniProt knowledge base is composed of 2 different data sets, SwissProt and Tremble. SwissProt contains records that are reviewed, which means that our teams of creators have read literature, and from it they extrapolate experimental data and add this to the proteins and to the records. And we can identify these records by this golden icon with a star. And at the moment, SwissProt has over 500,000 records. And on the other hand, we have Tremble, which is the set of unreviewed proteins that get annotation from automatic systems. And at the moment, we have about 250 million records of this. And you can identify this by this gray icon. So the types of information we provide in UniProt are very, very diverse. Here are some examples. So we provide the protein name, the function, description of what the protein does, but also put into context on a molecular level, cellular level, or tissue level. We can provide the taxonomy of the organism in which this protein came from, the subcell location of this protein, its 3D structure, domains and motifs, canonical or isoform sequences, many diverse cross-references to other external databases, etc. It would be great to have manual curation for all of these proteins to have a more complete database. However, some proteins do not have any experimental data. So how can we characterize those in that situation? Moreover, how can we curate the exponentially imported number of proteins coming into UniProt? With new technologies, there's a rapid increase on sequencing technologies, which allow scientists to sequence genomes not only faster, but with higher quality. And this results in genomes and proteins available, more and more genomes and proteins available to us. So taking this into account, manual curation is just not enough to characterize all proteins. So to address this issue, we use automatic annotation systems to broadly and reliably characterize proteins. So initially, when UniProt creates a new protein, it is unreviewed by definition. So these proteins reside in Tremble. If this new protein has experimental data available through scientific papers, then curators are going to pick this up and they are going to review it. And with this, with this new record, it's going to go from Tremble to SwissProt. But on the other direction, we use these reviewed SwissProts as the templates for automatic annotation in order to annotate Tremble. And we are going to look into this in the next slides. But as a summary, our goal with these automatic systems is to provide functional information to proteins that cannot be manually reviewed. So these automatic systems are reliable and traceable, as we are going to see next. So in order to propagate annotation automatically, we can start by thinking UniProt knowledge base as a space where many different proteins exist. So these proteins, they cover a wide variety of types of proteins that come from all different types of organisms. So firstly, what we do is group these proteins by similarity. So we are going to get proteins that are similar to themselves and group them together. Then what we observe is that inside these groups, we have a mix of proteins that have been reviewed and that have been unreviewed. So here on this diagram, we can see the reviewed ones being color-coded by orange, and the unreviewed are the blue proteins. So we can think of this a bit more abstractly, and we can say that each group is composed of a SwissProt space and a Tremble space. So a reviewed space and a non-reviewed space. So after confirming that each group has homogeneous proteins, and by that I mean that all proteins from a group have the same annotation, we can propagate that annotation from SwissProt to Tremble spaces. This is because all proteins from the same group are very similar. So each time we infer information this way, we can say that we created a new rule for propagation. And finally, we are going to remove groups that do not have any experimental evidences, since we cannot infer anything in these cases. So this is another way of visualizing what we just saw. In the first example, in group 1, we group these proteins together as they are very similar. If we know the function of the orange proteins, which are the ones that have been manually reviewed, then we can infer that information to the unreviewed proteins. Now, the types of annotation really depend on what curated data we have in the SwissProt. So in this first example, we are propagating a protein name and an EC number. And on the second group, we are propagating a function comment and a subcellular location. So this is an example of a rule in UniProt. We see that it's divided into two different parts. The left panel are the conditions, and the right panel are the annotations. So the conditions describe how we group proteins together. What do they have in common? So in this case, we can see that all of them have this PFAM signature, and they come from, they originally come from an organism, an eukaryotic organism. So we are going to see later what the signatures are in more detail. And at the end, the Tremble entries that met these two conditions are going to automatically get the annotations on the right panel. So they are going to automatically get this protein name and this subcell location, etc. So these are how rules work in UniProt. So just to show you how this looks at the end, if you access one of the Tremble entries in the UniProt knowledge base, you can see that this is a Tremble by looking at the left top icon. You can see a gray icon. So this is a Tremble, which means that it is unreviewed. And if you look into the function comment, you can see that it says required for normal Golgi function. And after that, you see a box saying to automatic annotations. So you know that this information comes from automatic annotation. And if you click on this label, then you can see specifically what are the rules that the annotation is based on. In this case, it's based on rules coming from UniRule and ARBA. And we are going to look more in detail on what are the difference between these two systems. But for now, you just need to know that these are two different systems that they coordinate together to provide a more comprehensive annotation to the protein. Now that we cover what a rule is, I'm going to explain to you in a bit more detail how we create these rules and how they are maintained. So we start by looking at our SwissProt space as we previously mentioned. So you can see here that this is a reviewed entry. This is a SwissProt entry, and you can identify by the gold icon on the top. So if you go to the family and domain section, you can see that the PFAM signature we used in the previous shown rule. So what we are going to ask ourselves, what are signatures, how we are going to use for the rules, how does this make sense? So to put it simply, a signature is a hidden Markov model, or an HMM, which is probabilistic models that describe functions on functionally a region of a protein. So an HMM can be small in length and can describe a domain of a protein, or can be large enough to describe the function of the whole protein. So the purpose of using the signatures in the rules is to group these proteins together so that we can infer information from SwissProt to Tremble. This is how we group. So these signatures are found through Interpro. So Interpro is a database that conglomerates information from 15 different smaller databases that classify proteins and provide these signatures to them. There are a number of signature databases that try to infer the protein function in different ways by using different methodologies, but at the end, Interpro gathers all these efforts and puts into one single database. And we are going to get these ones and map them into our UniProt accessions. So this is just an example on how Interpro displays signatures for a protein. So each colored bar here represents a different signature. The top horizontal grayscale represents this protein that has about 800 amino acids. And we can see on the right side the signatures of this protein. And this PFAM signature is the one that we used for the previously shown rule. So let's see how we create a rule using the PFAM signature we just looked into. Firstly, we need to see the proteins and how we group together. So if we go to the advanced search, and if you look into the type of information that we want to get, in this case, it's going to be the PFEM. And if you display there, if you write there the exact PFEM that you need, then you can search. And at the end, you will find that in your curator, there are 3,239 proteins that contain this PFEM. Now, some of them, 35 of them actually, are from SwissProt, so they are reviewed. 3,204 are not. So what we want to do with this is trying to, now that we group these proteins together, we're going to see the common annotations from these 35 SwissProt, and we can infer that to the other 3,000 proteins. So in this table, each row represents a protein and each column represents an annotation. All these three proteins have the same name, they have the same similarity comment, and the same subcell location. So the annotation overall seems consistent with all of these proteins. And because the annotation is consistent, then we are confident that the annotation from these reviewed proteins can be propagated in an accurate form into the unreviewed proteins with the same signature. And with this, we are able to create a rule. But before we do that, we notice that the function comment is quite different for fungi and non-fungi organisms. So all of these nuances are taken into account when creating rules. So this is the same rule we have seen before, but now we've taken into account these different function comments. As we've seen before, that would depend on the taxonomy of the protein. So if the protein comes from a fungus, it will have the first comment. If it's not, it's going to get the second comment that describes this kind of function. So this fine-tuning when creating these rules allow us to propagate information more precisely. And now that we've talked about the specificity of the rules, I would like to talk about the range of the annotations that we can provide. So we can provide annotations such as pathways, gene ontology terms, EC numbers, gene ontology terms as well, domains, subunits, information if this protein is part of a complex, etc. So all these annotations are going to provide you a clear picture on not only what the protein does in the catalytic activity, but also what's the context around it. And we are able also to add annotation from the sequences as special conditions. These are two different types of annotation being added here. So first, the bioinformatic software from transmembrane regions and signal peptide prediction is run on each of these sequences. And this particular protein has some evidence of transmembrane regions, so that that would be part of the annotation of this rule at the end. So the system that I've described so far is the UniRule, and we've seen that we, as curators, create and maintain these rules. However, we have other systems as well. So, for example, we have this one, which is the association rule-based annotator, or ARBA for short, and this is a fully automatic system. It also uses the manual annotated set of SwissProt as a training set. And it first it loads the data, and then it filters in order to create a highly reliable data set for training. And in the next steps, it creates the rules, it selects the most reliable ones, and it aggregates them in order to remove redundancy between them. And at the end, it also performs a cross-validation in order to ensure the quality through statistical analysis, and then it outputs these as a report. And in the end, it propagates prediction, just like UniRule, into Tremble. ARBA is a multiple class prediction system, which means it generates a model for all the annotations we previously mentioned. ARBA collects the exhaustive knowledge that is available in all data and selects only the rules that satisfy the statistical confidence criteria. And these rules are then subsequently filtered down to a small set of representative rules. So this means that we are filtering out rules to predict the same subset of proteins that other rules already predict to stop redundancy. And now that we covered individually how UniRule and ARBA works, let's just go through some of the differences and how these complement each other. So ARBA generates one annotation for each rule, whilst UniRule contains many annotations per rule. Another difference is that one protein can have multiple annotations coming from multiple ARBA rules, but it can only be touched by one UniRule. And at the moment, we have 25,000 ARBA rules and 7,500 UniRule rules. So you can see that they complement each other because one is more specific, but the other one has a broader range. One is manual created, while the other one is automatic. So all of these will provide better context to the protein when looking into it. In the UniRule rule previously shown, you can see many annotations contained in it. And here are just two examples where each rule from ARBA only contains one annotation. The first one corresponds to a catalytic activity, and the second one solely the name. So additionally, UniRule and ARBA provide sequence feature annotations. These annotate certain regions of the protein sequence. For example, they can flag where certain domain is predicted. Also, we can add SEM, which are sequence analysis methods, and this can predict the order regions of the protein sequence, for example, signal peak, coiled collets, etc. Some of these are going to be contained in the rules as well. And we can see here that they cover more or less half the tremble space that we have in UniProt knowledge base. So another system we introduced recently is the Protein Natural Language Model, or ProtNLM, that was developed by Google and validated by us. So ProtNLM uses a transformer model, which is trained on the protein sequence itself to predict the name. So at the moment, we use an ensemble of 7 different models to predict the name, 3 that analyze the protein sequence, 3 that analyze the organism taxonomy, and an optional one if we have an AlphaFold available to us, fold structure. So to put it simple, each model provides a score that individually is going to be calculated at the end to provide a final decision on the name. And this diagram represents the number of proteins in UniProt knowledge base. So the biggest large green circle represents the whole tremble with 253 million proteins, whilst the red circle represents the SwissProt, which only represents 252,000 proteins. So overall, UniRule and ARBA cover 57% of tremble, which means that tremble entries are at least being touched by one of these rules. And then if we take into account all of our automatic systems, then 87% of tremble is being covered by our automatic systems, which leads us to a very small percentage of our unreviewed entries that are not being touched by automatic systems. So the information that you are getting there is from the submitter itself, the submitter of the genome assembly. So we know that functional annotations are very valuable to the scientific community. So we are sharing this knowledge to the users, including our rules. So in order to share these rules, we have created a file format in which we can represent the UniRule and ARBA rules. And we call this the UniProt rule markup language. It is based on a well-supported and understood standard like XML, and it exposes the logic of these rules explicitly. To allow the users to use these rules, we have also created and released an open source software, a rule engine called Unifier that allows the user to use these rules that we created to annotate your own set of proteins. So the Unifier workflow is described on the diagram on the right. The inputs are the protein sequence, the protein taxonomy associated to the protein sequence, and the signature data provided by Interpro. Then the rule engine, using the different rules from UniRule and ARBA, it tries to predict all information for all of these proteins. And usually, Unifier requires several different tools and dependencies, but we already have these taken into account. So we created a container. So it's a container is an image that you can use through Docker, Singularity, or Podman. And this container already has all the dependencies installed within it, which means that if you download this container through our GitLab link, then you can use, for example, Singularity and run it right away without installing anything else. So in order to annotate your protein sequences with this unifier, the first step is to prepare the input data. So you need to prepare a FASTA file, not only with the sequences, but with a header containing the taxonomy of the sequences. It's important to say that the more specific the taxonomy is, the more reliable or the more specific the annotation will be. So you can have taxonomy on the family level or the order level, but that's going to be less specific if you actually add a taxonomy on the genus or the species level. So in this slide, it shows all the steps that you need to do to run Unifier in your environment. So in this example, we are going to use Docker, but you can use Singularity, for example. So first you need to check that Docker is installed, and afterwards you can clone our publicly available GitHub repository for Unifier, and this repository comes up with a script that runs everything you'll need. So the script provides a couple of options, but for most of the options, they are good default values, so you really don't need to touch many things. All you need to give is the command line. On the command line is the output file in the input file in the output directory. And with that said, you can simply run Unifier quickly to give annotations coming from UniProt in your own time, in your own HPC cluster or in your personal computer. So you don't need to come to our website for these types of annotation if you want to. So if you have anything you'd like to report back or provide us some feedback on rules that you believe that they are not that accurate, being because we need to adjust this or because you find something on a paper that contradicts what we have, then you can always give us feedback. So in our webpage, if you click on the top right icon here with the letter, then it will lead you to this page, and you can contact us directly, and we will assist you and potentially provide a better rule that we have nowadays. And if you would like to read further on our latest UniProt paper, we have this 2025 paper about our database that covers not only this, but many other aspects of our database. And more specifically on UniRule, we also have a paper on this. So if you are interested, I would recommend to give it a go and actually read a bit. And also we have a preprint on Google's ProtNLM as well. And with this, I would like to thank our teams and our funders that support us. So thank you. And if you have any questions.
Thank you. Thanks a lot, Pedro, for your webinar today. And thanks everyone for joining in. So we have a few questions already in the Q&A. How are entries prioritized for manual curation?
I guess this is a question
For me, entries are taken based on the current projects. And as you can understand that this is a very big database. So in case you are interested in specific protein, you may request the update of this protein. On every entry, there is a feedback tab where you can just write a message to us, suggest papers that can be used for curation, and we will have a look into that.
There's another question about, I think it's about citing a protein. I'm trying to cite a protein from UniProt, but the entry is incomplete or missing information. How can I obtain the necessary citation details for this protein regarding to publication information?
One thing on UniProt entries, there is a tab with publications. So from this tab, you can find the papers and information that was obtained from this paper. However, if you see that entry is incomplete and you know good papers that can be added to support certain type of information, just use the feedback tab and provide a feedback so we can update this entry.
How often do you revise the metadata of older entries? For example, potential DNA binding motif that the protein can bind.
So it really depends on the nature of where this information comes from. So we have rules. When it comes to rules, we have rules that are very old and other ones that have been reviewed very recently. But it really depends on this is on the rules, but also if we look into the SwissProt, there are some SwissProt entries that are quite old, and there are the other ones that have been reviewed very recently. So I'm not sure if you have a clear answer for all the proteins. Just one more thing. I just want to mention that if you have additional questions, you can always reach out to us. You can contact us through from our website, and we will answer you as best as we can.